This method can help answer key questions about biofilm development, such as how to identify key environmental conditions that affect biofilm growth and behavioral characteristics during development.
The main advantage of this technique is that the multi-channeled microfluidic plates allow for the quick acquisition of statistically significant results.
Though this method can provide insight into biofilm structure, it can also be applied to the study of antibiotic treatments and bioremediation.
Generally, individuals new to this method will struggle because careful attention to the details of the protocol and meticulous skills are needed.
To avoid an error in the connection of the instrument to the software, turn on the PC workstation first, followed by the fluorescence module. Make sure the fluorescence shutter is on, and turn on the hardware controllers. Next, turn on the imaging system controller and the CCD camera. Then, turn on the imaging station. When all of the equipment is ready, start the control application and enter the plate number located on the label on the side of the plate. For priming, first remove the 48-well microfluidic plate from the packaging without touching the glass surface at the bottom of the plate, and clean the glass slide at the plate bottom with a lens tissue. To prime the microfluidic channels, pipette 200 microliters of 37 degrees Celsius minimal medium into the output well, taking care to avoid bubbles, and place the plate onto the plate stage. Wipe the interface with ethanol, sealing it on.